All right, so our next presentation is going to come from Greg Bowman, Director and Associate Professor. He's going to be talking about the computational biology, crowdsourcing, and combating COVID on citizen scientist cycles. I think it's going to be interesting presentation here in terms of everything that's been happening with folding at home and what we've been dealing with for the past year at COVID-19. So welcome, Greg.
Thanks so much. I'll get my video working here. Give me one second. Okay, great. Well, thanks so much for the chance to join all of you. This is a lot of fun to hear about all the cool advances, and I'm looking forward to the rest of the day. So I guess I'm a little bit of an outlier in that, you know, I'm very passionate about computers, but my driving passion, if you will, is understanding proteins. And so just for some background, for those of you who haven't thought about proteins for a while, these are the molecular machines that are responsible for most of the processes that we associate with life. So muscle contraction, that's proteins moving, your eye sensing light, that's proteins undergoing a change in their conformation or shape in response to light. You know, your hair is largely protein. Proteins are what breaks down your food and builds new cellular components. So they're busy little machines. And like the machines that we're used to working with on macroscopic scales, they have lots of moving parts. But one of the challenges is that, you know, unlike machines on macroscopic scales, we're generally blind to what those moving parts are. So in general, for many proteins, we either have, you know, no idea what shape they adopt, or we have like a single snapshot of what they look like. So what I'm showing here is a protein that we think about a lot. It's called beta-lactamase, and it is expressed by bacteria, and it actually binds to and degrades antibiotics like penicillin, right? And so the role of this thing is to protect bacteria from our drugs that are meant to kill bacteria. And so there's a lot of interest in understanding how these things work and designing ways to shut them off so that we could, say, restore the efficacy of our existing antibiotics. So what you're looking at here is a single structure of what this protein likely looks like much of the time, where each atom is shown as a little sphere colored by what chemical element it is. And what you can see is that this is a pretty compact shape, and I've labeled this pocket in the surface, which we call the active site of the protein, because that's where it does the active thing that it does, right? In this case, breaking down antibiotics. So there's a huge amount of information in these structures, but really it's just the tip of the iceberg, right? Because every bond between a pair of these atoms is capable of rotating. And so there's a huge number of degrees of freedom, and, you know, essentially infinite for all practical purposes, number of different conformations that a protein can adopt. And there are cases where, you know, the single structure is so much more stable than every other structure that it's reasonable to say that this one structure we look at is the structure of the protein. But for most biological systems, I don't think that's the case, and there's actually a lot going on. And one of the issues, though, is that because of the size scale that these machines exist on, you know, there's no microscope where we can just zoom in on these things and watch them in action and say, oh, okay, you know, that's how floppy it is, or that's how it works. And so one of the things that I've, you know, dedicated my career to is using computer simulations to watch how every atom in a protein like this one moves throughout time. And start to understand how these amazing machines work in silico and see what utility we can derive from that. So here's just a little snippet of a simulation. You can see all the atoms wiggling and jiggling around stochastically. And here, where I've labeled cryptic pocket, is something that's really fascinating to us, because you see this relatively large scale motion compared to the jittering going on elsewhere, where this large cavity opens up that's comparable in size to the active site. And this is really exciting, because if you just have that static structure I showed you before, the only way that's obvious to drug this protein is to design a small molecule that binds tightly in the active site and prevents this thing from binding to antibiotics and degrading them. But when we find sites like this, which we call cryptic pockets because they're absent in those experimental structures, these could present exciting new opportunities, both for understanding how these machines work and in this case, for throwing a wrench in the cogs and preventing them from working. Because what we found is when this pocket opens up, it also causes subtle changes in the active site that turn this protein off. And so we've actually used this structural information from our simulations to design small molecules, drug-like molecules that we've then shown in subsequent experiments actually bind to this part of the protein and turn it off. So this is a really cool way to potentially manipulate these biomedically important systems that we just wouldn't have had any idea about without the sorts of detailed insights that we can get from these simulations. All right, so this is extremely powerful, but now to come to the challenge, right? So these simulations are extremely computationally expensive. We typically have anywhere from 20,000 to 1 million atoms in our simulations. At each time step, we're calculating, the most brute force method, at least, you'd have to look at the pairwise interaction between every pair of atoms in the system and calculate how they're going to push and pull on each other. And if you were able to say, okay, well, where is each atom a second from now, that wouldn't be so bad. But in order to prevent unphysical things from happening, like two atoms, jumping through each other because you propagated that velocity too far forward in time, we need to take very small time steps on the order of a femtosecond. And so, getting up to, the sorts of seconds or timescales that we might be interested in is 10 to the 15th iterations of this non-trivial energy calculation. So the upshot of this is that on a typical gaming desktop, for example, you're likely to be able to do tens or hundreds of nanoseconds of simulation per day, but we want to be able to get to like 6 to 12 orders of magnitude slower timescales. So a lot of computation could easily take hundreds of years for easy problems to age of the universe for some of the harder problems. And so what we've done is take a very different approach to this problem, which is to say, okay, well, instead of thinking about this simulation as atoms moving as a function of time, we're instead going to think about it as what we call an energy landscape. So the idea is that you have this high dimensional landscape, just showing a cartoon of here. Each point on this landscape, you can imagine, corresponds to some protein conformation. And when we watch one of these simulations with the atoms moving around, effectively what we're watching is a biased random walk on this surface, where the protein is spending exponentially more time at lower energy structures down here than at progressively higher energy structures like the guys up here. Right. And this is extremely powerful because now instead of saying, okay, well, we need to make our one simulation run faster, what we can do is say, okay, well, really, we don't need that one simulation to be faster. Really, what we need to do is build a map of this space. And we do that with an approach that we call Markov state models, or MSMs. And so the idea is that we're going to run a bunch of simulations, and we're going to use those to infer where the minima in this landscape, which are going to become roughly nodes in this network, are, and what the transition probabilities between adjacent minima are. Right, and so this is an extremely powerful framework for a number of reasons, but if you think about a more familiar problem like wanting to build a maps app of the United States, for example, right? You could say, okay, well, I want to get information about all the roads as quickly as possible, so I'm going to build a Formula One race car. I'm going to make this one car as fast as I can by paying a lot of money, and then I'm going to zip around the country as fast as I can to find all the roads and make my map, right? And that's not necessarily, that would work, right? But you could get the same information far more quickly if you instead said, okay, I've got this big space. I've got a whole bunch of people throughout this space that have, their CRVs and their, Mercedes and their, whatever, that are far more affordable than that Formula One race car. And I'm just going to have them send back their GPS coordinates as they commute to work and go to the grocery store. And now on the server side, I'm going to take all of that data and based on overlap between the regions of space they've explored, I'm going to build a map of this space that no individual ever explored on their own. And so that's the approach that we take to taking a seemingly intractable problem that is, unapproachable with, a single monolithic calculation and break it down into trivially independent pieces that can be performed on independent computers. And so with this framework in mind, we of course said, okay, well, this is cool. We can run a bunch of simulations on our local HPC, for example, but it'd be nice to have more. And the NSF and NIH aren't going to give me a Summit supercomputer to myself. So what we, and particularly Vijay Pandey, formerly at Stanford did is start the Folding at Home Distributed Computing Project. Right, and so the idea is to invite anyone with a computer and an internet connection to volunteer to become a citizen scientist and run these snippets of simulations that we call work units on their personal computers. And so this is just a map of the globe with a little pinprick of light everywhere that someone is volunteering to contribute their compute power to this massive globally distributed compute network. And this has been super cool and provides some really interesting features, right? So the way this works, just to walk you through it, is that someone installs a piece of software on their home computer, for example. We call that software the client. On our side, we have a bunch of machines that are essentially decent machines with hundreds of terabytes of storage space that we call work servers. And then we have a load balancer called the assignment server that sits on top of those work servers. And the work servers are also all around the globe because there's about half a dozen research groups around the globe involved in the project. And so what happens if you're at your home PC, you spin up your client software, and what it's going to do is reach out to the assignment server and say, here I am. I've got a GTX 3080 because I was lucky and snagged one. And such processor, and I'm available to do work. The assignment server will then match it with the subset of simulations that are on the queue that are appropriate for that hardware. And it's going to send them the starting confirmation and parameters for one of these simulations that we call a work unit. And we target that those will take on the order of an hour for a typical machine in the network. So now your client software is going to go run this simulation in the background, over and over and over again, calculating how each atom pushes and pulls on each other atom, and updating their positions. And after running for fixed amount of simulation time that we've specified, it's going to send back this work unit, which is a series of snapshots, kind of like a movie, where we stored the protein conformation at regular intervals in time. And then on our end, we've developed a bunch of algorithms for taking all of this data and building one of these maps. And so, and I'll just mention, so the work units, there are a fixed length. And so what happens when someone sends back a work unit is that the work server does some basic checks to make sure it looks like simulation data, and then takes the last snapshot on that movie and adds it back to the queue as the starting point for a new chunk of simulation. So we can get much longer simulations effectively than a single work unit. And then it awards the volunteer points in proportion to how hard the calculation was and how fast they did it. So there's a cool social science aspect to this too, with gamification, with the point system and whatnot, and the whole ecosystem that's built up around this. There's cryptocurrencies that are tied to our point system, and people who will do detailed studies of what hardware to buy to get the best bang for your buck. And it's really cool to see all of this grow up and all the around our science, and people's enthusiasm for the science and interest in understanding how is it that you're tackling Alzheimer's disease, or how is it that you're tackling COVID-19. And maybe of interest to you, it gives us some interesting features like a sort of fault tolerance that is very appropriate to large scales, because in the end we don't we care if someone stops participating in the network, but in the sense of we don't care that because it doesn't hurt our results. So if someone's machine dies or they decide not to participate anymore, we get slowed down an iota because we lose whatever little bit of simulation they had actively going on their computer, but in the end, we still have most of the data coming back from all of these independent machines. and then we're doing this inference of this map server side. And we have some cool things that we've done this with this now in the direction of active learning, for example, through a methodology we call adaptive sampling, where what we do is we run a batch of simulations, we get all the data back, now we build one of these maps, and then we use that map to decide, well, of the conformations or protein structures that I've discovered so far, which would it be most valuable to start the next batch of simulations from to improve my model as efficiently as possible, both in terms of exploring new regions that I haven't mapped out yet, as well as improving the estimates of these parameters like transition probabilities. So reducing the uncertainty in key model parameters. So at the beginning of the pandemic, we were working on all kinds of different problems. One of the cool things about this is that it really is a platform that can be taken in many different directions. So in my lab alone, we were working on Ebola virus. We were working on Alzheimer's disease. We were working on, one or two forms of cancer, all kinds of interesting different proteins. And then, of course, about a year ago now, it became clear that this SARS-CoV-2 virus was going to become a pandemic. And so what we did is say, okay, well, we have this very general platform that we can take, basically in any direction. And so now it looks like we should be taking this in the direction of COVID-19 work. So we very quickly pivoted to, focusing all of our attention on this one set of related problems. And at the end of February, we launched our first simulations of, one of the about 30 proteins from the virus, with the intent of starting to understand, how this protein works and how we might be able to use that information to inform the development of therapeutics or vaccines. And the response was really amazing. So at the beginning of 2020, we had about 30,000 devices around the world that were participating in folding at home. After we launched that first set of simulations at the end of February, we had a huge spike in participation in folding at home. So in blue here, I'm showing the number of cumulative downloads of our client software, that software that runs on people's personal computers. And then in orange is, growth in number of cases of the coronavirus with some of the key events marked. And so what you can see is that, within the first couple of months of the pandemic, we went from about 30,000 devices to well over 1 million devices actively participating in folding at home. And so this was a really incredible ride, right? At the, in 2019, we were already, running calculations that would have cost millions of dollars on the cloud on a routine basis with these 30,000 devices. And so with the massive growth in this network, we had quite a lot of compute power on our hands. So here I'm just showing, estimate of peak performance of some of the fastest traditional supercomputers and a very conservative estimate of the total throughput of folding at home. And so what you can see is that, even with this very conservative estimate of what was available to us, we were the first to reach the exascale and had at least 5 times the peak performance of what at the time was the fastest supercomputer in the world, the Summit supercomputer. And so that's really opened the opportunity to do a lot of science in parallel. We very quickly also were sprinting to, figure out how to get server-side infrastructure for folding at home working on the cloud. So that was an adventure too, because of course, it takes time to get grant money and buy new hardware. We've been running everything on-prem, and we handled the first, five or so fold growth without breaking a sweat, which is, pretty impressive, I think. But as we got to 10-fold growth and above, it was starting to bring the system to a grinding halt. And so, it was great to engage with a number of companies who helped us get on the cloud. And so what we did with all of this is say, okay, well, in a normal paper, one focuses in on one protein and gets something like a microsecond of simulation data. That's a pretty standard, good data set in the field that can result in a nice publication. But we had so much compute power that what we actually did is start running simulations of every protein component that we could from SARS-CoV-2 as well as related coronaviruses and some of the important host factors that they interact with and variants of these proteins and some of the complexes where two or more proteins bind to each other, so that we can try to figure out how as many of these things as possible work and get that data out there for others to work with also, because this structural data can be a rich source of information for accelerating, drug development and other medical research. And so one of the things that we initially focused on is this complex of three proteins called the spike, which are all of these protrusions that you see in red. And these were really fascinating to us, right? Because when you, the spike complex is the component of the virus, which is the first point of contact between the virus and a potential host, a human cell. And so what the spike's job is to grab onto a protein called ACE2 that's present on the surface of many human cells. And then it undergoes a dramatic change in its shape or conformation that triggers the virus to insert itself into that host cell, where it can begin making copies of itself and eventually burst out of the cell and go off to hunt for new prey, right? So this becomes very interesting for two reasons. One, the spike plays such an important role. So if you can block it from engaging with a host, that would be a great therapeutic, for example, or if you can mimic it, that's one of the primary ways to develop a vaccine. But it's also interesting because being on the surface, it's exposed to our immune surveillance mechanisms and to therapeutics that humans have designed. And so the coronaviruses have evolved a number of really clever defense mechanisms. And one that fascinated us is that when you see these spike complexes, like in this canonical image of the virus, the spike is actually closed up on itself in this structure, right? And so the surface of the spike that is actually responsible for grabbing onto this ACE2 protein and initiating infection, it's actually buried in this closed up structure. And so what that does is, kind of like a turtle pulling its head into its shell, is that it allows the spike to hide from being recognized by our immune surveillance mechanisms. But just like the turtle, it can't stay in its shell all the time where it's nice and safe. It has to poke its head out every once in a while in order to be able to eat, for example, right? And so to engage ACE2 and initiate infection, the spike has to open up, and then it's able to grab onto ACE2, and a whole bunch of events come after that initiate infection of the host. And so what we wanted to do was see what this opening event looks like, right? Because people had solved closed structures, experimentally solved closed structures of the spike. So here I'm showing an example. Up in the upper left here is where the spike is tethered to the surface of the membrane. In the lower right is the receptor binding domains, or RBDs, that are closed up on themselves to protect the ACE2 binding interface. And these things somehow have to open up to expose that surface. But it really wasn't known what that looks like or what therapeutic opportunities it might present. And so for us, this is a big system, right? When I finished my graduate work in 2010, it was an exciting advance that we'd gone from simulating 10 residue peptides to 100 residue proteins, residue being one of the chemical components of a protein. So the spike here is 3,600 residues. So a couple of orders of magnitude, an order of magnitude bigger, and a much bigger simulation, because we have to stick this in a three-dimensional box of water. So every time the protein you want to study gets bigger, the box of water becomes even bigger, and there's more water and other stuff in the water that you need to simulate. So a much more expensive calculation. And so many people got interested in simulating the spike and trying to contribute to this. The longest simulation that anyone else has presented is 10 microseconds of data. We have well over a millisecond of data for the SARS-2 spikes, so 2 orders of magnitude more data. In addition to looking at that spike, we also have a similar amount of data for the spikes from 2 or 3 other coronaviruses, so that we can understand what's common to them and what's different that makes SARS-2 special, so to speak. And the rest of the proteome, we have like a millisecond of data for each of those. So in aggregate, we have about over 0.1 seconds of data, which is just amazing given that, like I said, 1 microsecond of simulation is, a good data set in the field. So like 6 orders of magnitude more data. All right, so here's a little bit of what we saw, right? So at the beginning, things are just wiggling and jiggling. That's all you see if you've got 10 microseconds of simulation. But with a millisecond of simulation and our clever sampling algorithms, you see these huge conformational changes where one of these receptor binding domains really peels away from the rest of the protein and exposes all of these buried surfaces. So this part right here is what actually binds to ACE2. So that we knew had to get exposed, but we didn't know that all this stuff back here had to get exposed. And so that's cool, right? Because these are now new drug targets that you could consider that you wouldn't have considered if you thought the spike just sat in that closed conformation and didn't undergo these sorts of structural changes. So this is like our cryptic pockets, and that you wouldn't expect these surfaces to be exposed. And indeed, since we publicized our work, a number of antibodies have been developed that turn out to bind to these deeply buried surfaces. And that just wouldn't make sense without the sort of structural insight we have, but makes perfect sense once you see simulations like this. So super exciting, and now we're using the same sorts of technology to go, look at these emergent variants, for example, and try to understand how they modulate the relative probabilities of open and closed states, because that's an important determinant of how well the spike can hide from our immune systems, as well as a trade-off with how well it can infect a host by binding to this ACE2 protein. So super exciting biophysics. Right. So there's a whole bunch of other proteins that we've delved into. We've also made all the data publicly available because we just don't have the throughput to go through all the data and make sense of it on our own. And so we're hoping that others will dive in and, take a stab at different parts of it. We are working on a number of the systems and asking questions like, how do some of the mutations that we're seeing, affect the behavior of these proteins and potentially the pathogenicity of the virus. So just to give one more example here, we've been looking at another protein called the spike, called NSP16. This is another important piece of the virus's immune surveillance mechanisms. In this case, this protein binds to RNA that's made by the virus here, another molecule called SAM, and it transfers a couple of atoms from SAM to the RNA to put a little cap on that RNA. And this is important because all of our human RNA has that cap, right? And so without it, our immune systems are really good at recognizing uncapped RNA and saying, there's a virus or something here, quick guys, scale up the army and let's kill it. Right. And so what the virus is able to do by having this protein called NSP16 is add a cap to its own RNA, which is like a signal that this is self, and immune surveillance mechanisms say, oh, no problem here. And so, the virus can kind of act like a Trojan horse and continue about its business without being bothered by our immune system. One of the cool things here is that despite being essential to the virus's ability to replicate, this is a really hard drug target, right? Because our cells also have proteins that are similar to NSP16 that are responsible for capping our own RNA. So if you design a drug that targets these obvious binding sites where the protein binds RNA and this molecule SAM, what happens is that you get a potential drug that kills the virus, but it also makes people really sick and maybe kills them because it shuts off our proteins also. And one of the things we observed, though, about the viral protein is that unlike the human protein, it is only active when it is bound to another viral protein called NSP10, right? And so this is cool because it suggests that through a process we call allostery, somehow this part of the protein over here is communicating with this part of the protein over here such that the protein can only do chemistry in these pockets when NSP10 is present all the way over here. And the fact that there's this communication means that you might be able to design a drug that does something over here to the viral protein, but maybe has no effect on the human protein, for example, and presents a much better candidate for drug development. So we ran simulations of NSP16 and alone, and also this complex with NSP10. We got tens of millions of protein structures for each of these. And then the question is, okay, well, what do you do with all of that data, right? Because you can't just look at tens of millions of structures and say, here's what happened. And so we've developed this deep learning approach that we call DiffNets. And I'll try to be very fast here, but you're probably familiar with autoencoders where you take some high dimensional input, you coarse grain it to a much smaller number of variables, and then you decode it and try to reconstruct that original input. So we took this paradigm and said, okay, well, this is good. If we input a protein structure and train the network to minimize the reconstruction error, we're going to get a coarse graining of our data that is very good at recapitulating the geometry of the protein. And so that's valuable, but it doesn't necessarily help us figure out how these monomer and the complex of proteins, how they differ. And so what we did is we added two other elements. One is a supervised component, where this network also has to be able to predict some biochemical feature of different ensembles. So it has to be able to predict, say, that the monomer, the single NSP16 protein is inactive, but it's active in the presence of NSP10. And then the last ingredient is this expectation maximization scheme, where we're actually updating those labels on the fly to learn a self-consistent labeling of our data. So really cool stuff, but just to wrap up, the upshot is that we find some really cool conformational changes that distinguish the monomer from the complex and explain how this thing is activated. And even more importantly, we find this cool cryptic pocket over here that is present in the NSP16 proteins from, it looks like, all of the existing coronaviruses, and is absent in the human proteins that perform the same function. So this is a great candidate for developing a pan-coronavirus inhibitor that doesn't affect the ability of our own cells to survive. So with that brief summary of all the data, like I said, usually people have a microsecond of data for one protein. We actually have over 100 different systems now that we've studied, over 0.1 seconds of data. This is very compute limited, so we rarely have to worry about storage space, but in this case, we've got over 200 terabytes of data, and we've made all of this available online so that other people can work with that. And so with that, I'd like to thank my group, as well as everyone who's volunteered to help with Folding a Home and the other 5 research groups that are involved in the project, all of our funding sources, and all the companies that really reached out to help us as we struggled to keep up with the influx of participants in Folding at Home and to make effective use of all of that data. And so to wrap up, I'll just say we've also got through a collaboration called the COVID Moonshot, we're putting this to practical use, and together have developed a small molecule inhibitor that is now in animal testing and is showing promise as another way to combat the virus. So we're going all the way from the very basic stuff and really cool computing algorithms to something that hopefully will go into people in the next 6 months or so. So with that, I'm happy to take a few quick questions if we have time.
Thank you, Dr. Bowman. Yes, we have a couple of questions. In terms of time intervals, what time intervals do you use between snapshots of the simulations?
Yeah, so typically so each step of the simulation is on the order of a femtosecond, so like 10 to the negative 15 seconds, and we're usually storing confirmations every about 10 or 20 picoseconds.
Okay, and then the second question was, is each job run twice to protect against soft errors?
So we don't run each job twice. One of the cool things with this is that usually we're running anywhere from 1,000 to 10,000 simulations in parallel. So if there's an error in one, it really doesn't have that much of an effect. And if it's a bad error, it's easily recognizable because the protein just explodes. And so we just throw that data out and we still have most of the data and we can move forward with that.
Got it. I think I've got another one here. Have you used FAH, Folding at Home, to simulate in mass how mutations will affect virulence?
Yes. So we are doing that now. So we have simulations going of the spike complexes from the UK and Brazil and some of the other concerning variants. And one of the cool things about this is that it's time consuming and expensive to do experiments, right? But we have the potential to automate our pipeline and have this be a first pass screening of new variants where we can make some inferences about whether that variant is something that we should be worried about or not. And, in the long run, this is a great opportunity for precision medicine because then we can take that data and say, okay, well, how do we have drugs that work on this variant versus that variant or whatever? And that has lots of applications in human health.
Let's see. The folding occurs in simulation in a box of water. Any study of what happens to the protein if the water is moving/changing? Is COVID more active from a cough or from a non-moving surface?
Oh, yeah, great question. So the water is always moving and changing in the simulations, and we can get at questions like, well, what happens if you have different stuff in the water, like different salts or other small molecules are present. So these sorts of questions are all things that hypothetically can be addressed. In general, we have a pretty standard set of what goes into that. That is a reasonable mimic of what lots of proteins see, and so we haven't delved into that in the case of the coronavirus.
And last question: what protein force field are you using? For example, amber.
Yeah. So we use a couple of different force fields. For most of the work, we actually use a force field called Amber-03. For those that aren't familiar, the force field is just the mathematical description of the interactions between different types of atoms. So what are the charges on the atoms and how do they push and pull on each other, and how strong are different bonds, and so on and so forth. This is one of the really cool things is that lots of the modern force fields that have been developed within the past 20 years work quite effectively if you have enough compute power. And so, there are newer force fields which hypothetically are better, and there's room for improvement. But Amber-03 has been very good to us in terms of being predictive of the experiments that we're doing in our own lab.
Okay, perfect. Thank you, Dr. Bowman. Appreciate the presentation.
Thank you. My pleasure.